Okay, hello everyone. Thank you for joining us for the October 5th live stream. For this week, I wanted to go over the special live stream that we had last Monday with Ed. He's in the comments, or he was at the beginning under Patrick, and he's gone by Glenn before. So after that live stream, he started posting stuff that were just so hilariously scientifically wrong. I wanted to have a little fun going over that, and hopefully maybe he will learn something. I can tell in the comments that he already has kind of shifted what he's saying. So now he says a peptide bond is a covalent chemical bond linking two consecutive alpha amino acids, C1 and N2. Before he didn't say alpha amino acids. He was calling the C1 and N2 alpha C1 and alpha N2. So maybe he did learn something. Who knows? I really doubt it. But what we will kind of go over a list of different things he said, and kind of explain why he's dumb. And we'll also talk about kind of what we want to do with him in the comments. He claims that I'm blocking him. I am not. And I'm not reporting him because when he finally kind of figured out that if I had blocked him, all his comments go away, and that's obviously not true. So he's now saying that it's because I reported him, and for some reason, and reporting him gets rid of the comments, but for some reason I didn't report all the other kind of stupid things he said. I only quote unquote reported I don't know, some other stuff that he thinks that is not showing up because, I'm deleting it. So,
But it hasn't
so
said
we'll have
anything
that.
that breaches YouTube TOS as far as I can see.
Well, if it did, it wouldn't show, right? Because
Yeah.
YouTube would have removed it. Yeah. So
Logic's not his strong point.
yes, it definitely isn't. But on the stage, we have Ook, who just spoke, and Blarg and Tuna Man. Hello, guys.
G'day all.
Good day.
Hello.
I already said
Hello.
hello. You don't get it twice.
Yep. All right. So I did send people the list, but I can go ahead and put it up so that people can follow along. I have to try to make it bigger. So he has a lot, and also this is not comprehensive, of course, because he's said a lot of dumb things that might not be on this, but we should be able to go through this relatively quickly. I was hoping Neil was going to join. I guess Neil has education in organic chemistry, I think, and I actually believe him when he says that.
Yeah,
Unlike
it was organic chemistry. And
okay,
I'm just going to pop out and log back in because I'm getting an echo through my thing.
okay,
All
right. Yeah, and so I believe Neil, but I do not believe Ed when he says that he has a biochemistry degree and a minor in he has a microbiology degree and a minor in biochemistry. And he had said he had education in biochemistry before, and whenever asked about it, he wouldn't specify what that means. But when he had that interview with Dr. Dan, Dr. Dan did say that he said, oh, okay, so you have a degree in microbiology and you have you minored in biochemistry. Is that right? And then he said, yeah. So he confirmed it. And when I asked him what exactly was said to make that comment to Ed, he actually didn't know, he didn't remember Ed actually specifically saying that. that he has a degree per se. I think he said something vague about how he has education in those. Oh, is it coming from me? He said something vague about how he sorry.
Yeah, yours was the only.
Yeah, it must be yours.
Yeah, yours was the only one unmuted.
Sorry.
Yeah, yours
Oh,
was the only.
that's
Yeah,
interesting.
it must be yours.
Yeah, yours was the only one.
Oh,
Unmuted.
okay. Let me know if the echo, or is there an echo even when I'm not talking?
Yeah, yours was the only one.
Oh, okay. Let me
Test,
know if
test, test.
the echo, or is there an echo even when I'm not talking?
It's running, the echo seems to be about 10, 15 seconds behind when you speak. It's running. Cue. The echo seems
I
to be
wonder it's because I have.
10, 15
Oh, I know why.
behind when you.
What you guys are hearing is the stream, the live stream. I had the tab muted, but I think it was still showing up. Okay, how's that?
Perfect.
Okay, all right. So in the when I asked Dr. Dan, he basically said, "Well, Ed didn't specifically say that he had a degree in microbiology and a minor in biochem." He kind of alluded to having those educations, and I think Dr. Dan sort of took that as to mean that he has a degree. But when he said that during the stream, Ed did not say, "No, you're wrong. I don't have a degree." He said, "Yes." So he's claiming, or he has claimed, that he has a degree. But everything we see him post pretty much says otherwise, that he absolutely doesn't know any of this, know any biochemistry, definitely, and he definitely doesn't know microbiology. So going through these should help kind of let people understand why we think that way. So um, unless any of you guys have anything else you want to say, I can start with this list. Or if you guys have anything you want to add to that.
Nah, get into it.
Go.
All right. Okay. So number one, he thinks lipids are fats. And this type of error he makes often, where he confuses the member for the set and the set for the member. So lipids are not fats. Fats are lipids. So there are different types of lipids, like triglycerides, which are fats, and then there are phospholipids, which is what we were talking about when we were talking about micelles. So this is where something he doesn't understand. It's minor, but he keeps on saying that while calling people dumb or stupid or brain damaged. So it's just kind of funny when he himself isn't correct when he says those things. And if any of you guys have anything to add to this, just let me know. Some of them are funnier than others. The other one, he thinks peptide bonds are fragile. And this is the rationale he used to say that abiogenesis isn't possible, because peptide bonds would just automatically break up. I'm not sure if I go through him talking about hydrolysis. It might be later, but I might as well talk about it now. So I had mentioned that the peptide bonds are not fragile, and if he even took a little bit of time to look it up, he would tell. He could quickly find out that he's wrong.
The dead giveaway, actually, for me, is that when you digest protein, just going through acid in your stomach is not enough. You need a protease enzyme to break the things up. You need something extra. It just is not enough to break it by itself. And that tells me that the stuffs must be reasonably tough.
Yep. And the half-life is, hundreds of years to maybe 1,000 years in liquid and at room temperature. So I told him when I brought up the half-life, shoot, I have to find what he said, because it was so obvious he didn't understand what I meant by half-life. So he made a really stupid comment about the half-life. Oh, here, I'll share this. So here I made a comment about Half-life of a peptide bond at 25 degrees is between 350 and 600 years. I've seen like some say higher, but anyway, it's hundreds of years. And his response is that QC lost again. We're not talking about the half-life of peptide bonds. We are talking about half-life of peptide bonds as a measure of how stable that bond is. And I get the sense, and I don't know for sure because he didn't kind of repeat this, but I think he was thinking half-life as in like atomic half-life. I don't know if you guys like have other thoughts on that.
Just I'm not sure whether he was thinking that direction. Probably because I suspect he hasn't come across the term before.
Mhm.
My understanding, though, would be that this half-life thing makes a whole lot of sense because when you think about really large, complicated proteins that might be thousands of units long, their lifespan is not as long because of the half-life per bond.
Yep.
As opposed to relatively simple ones, which would be basically just about bulletproof. And that's what we're talking about in abiogenesis, not the giant complicated ones that have evolved in the billions of years since.
Right. So as you mentioned, you need a protease to essentially, catalyze that reaction, because you could just like put it in very high temperature or you could put it in acid or very strong acid or base. Those are not enough to actually break those bonds. You would have to boil it at very high temperatures in either acid or base for a long time for it to break, or have a protease to do that. Now, interestingly enough, and the way that the bond breaks is hydrolysis, in the sense that a water molecule essentially have to get in there for you to break up that peptide bond. But the funny thing is, like, he basically says that it's not hydrolysis that breaks up the peptide bond. He's saying that it's the water, meaning that the water is physically breaking the bond. So here he says, no, it's not hydrolysis. He's saying that hydrolysis isn't a breaking of the bond. Ha ha ha, clueless. And he says other times, multiple times, where he talks about how
Hang on.
So yeah.
Just for me, is he saying what the movement of the water breaks the bond?
I think he's saying that the force of a water molecule hitting that bond is what's breaking it up. I think that's his conception of how that works. He's saying that, hydrolysis is a chemical reaction, and it's not a chemical reaction that breaks up that bond. It's the water molecule. So,
okay.
right here, if he says it's the physical force of water molecules that break the bonds, not a reaction. So he's saying it's not a chemical reaction that's breaking the bond. It's like, billiard balls where a water molecule is.
Exactly what I was picturing. It's sort of like, bang, bang, bang.
Yeah.
All right. So I got it right. Okay. See,
So,
I'm learning.
so that pretty much says that he hasn't learned any biochemistry. Hydrolysis is a fairly common reaction that you learn pretty early on in biochem. Hydrolysis and condensation reaction. But, for him to think that the breaking the bond is this physical force of a water molecule, it just astounds the mind that he claims to have biochem education and thinks that this is how things work at the molecular level. Okay. Anyway, so I'll go back to the next one, because although I'd like to just kind of spend some time on this, I don't think we have time to go through all these in detail. Okay, and then so anyway, that's the reason why he's saying that abiogenesis is impossible, because he thinks it's so fragile. Oh, I do have this thing later. He thinks that peptide bonds are broken through physical contact with water, not hydrolysis. So we covered that. And then the other thing he spent a lot of time about was calling peptide bonds, and he's calling them alpha bonds. And he keeps on talking about how, of course, these are alpha bonds. And he like went after Neo quite a bit on this whole alpha bonds. Oh, he says you can Google peptide bonds, and it says alpha bonds. So let me just show you the results of what happens when you Google peptide bond. So what you get is basically an excerpt from Wikipedia. And here it says a peptide bond is an amide type of covalent chemical bond linking to consecutive alpha amino acids from C1, which is a carbon number 1, of one alpha amino acid, and N2, nitrogen number 2, of another along a peptide or protein chain. So what I think what he's getting confused is this alpha part, that because it's an alpha amino acid, that the bond, any bond between the two, whether it's a peptide bond or it could be some other bond, they're just alpha bonds. And he thinks when people say alpha bonds, people know that you're talking about the peptide bond between two amino acids when you say alpha bond. As opposed to when you say peptide bond, everyone does know exactly what that bond is. You have no idea what that person would mean when they said alpha bond. So another example of how he couldn't possibly know biochemistry.
Kept calling the N2 bond, the N2 nitrogen, an alpha N2.
Yep.
As
Yep.
well. You just go, I don't even have a chemistry background, and that's obviously bullshit.
Yeah, I think he just like thinks that it's all alpha, or he can just call it alpha because he's saying the bond is alpha. So all the molecules or the chemical connection is an alpha connection, or is an alpha element. So let me see if I can find where he actually says something this stupid. So here, we're wrong about proteins being produced outside of cell, and wrong about alpha bonds not existing. So we didn't say alpha bonds don't exist. When I actually look up alpha bonds, it's talking about some glycosidic bond with sugars. But within amino acids, there are no such thing as an alpha bond. But he says that alpha bond exists between alpha C1, like you said, to alpha N2. And that is how amino acids bond. So he doesn't say anything about the bond itself, that the molecules are somehow bonded through this alpha bond, but he can't say really anything about that bond. Well, except I guess he does say it's alpha C1 and alpha N2. So the alpha amino acid is called an alpha amino acid because the side chains and the carboxyl and amine and all that, they are all connected to the alpha carbon. So that carbon in the middle amongst all that is what the alpha bond is. So let me see if I can. I thought I had the picture of this. But yeah, so that carbon is the alpha carbon, but the bond is not between that alpha carbon to the nitrogen. That C1 carbon is not the alpha carbon. It's actually C1 basically means it's like one I guess bond away from the carboxyl um, the carboxyl group. So the C1 is actually, oh no, so I said that wrong. I'm trying to bring up the picture so I can show you guys what I mean. Of carbon. Okay, so maybe this will work. No, this won't work. Oh, I didn't mean to do that. Need to make this bigger. I guess this picture will work. Let me share it. Sorry about this, guys. Oops. All right, I'll just share this. It's hard to tell. It'll be hard to kind of see, but what is showing up here is, oh, okay, it's still showing up. So this is basically the peptide bond. Let me see. So this carbon is the alpha carbon, and as you can see, it's denoted with the I don't know how I can get this picture to just show up. Okay, here we go. This should work. Oh, okay, here we go. All right, so this should be better. All right, so this carbon with the alpha, so that's the alpha carbon, and this and so this amino acid is an alpha amino acid because the side chains, amino group, carboxyl group, and the side chain itself, they're all connected to the alpha carbon. There are beta amino acids where the side chains are on a different carbon or different place in the acid. But anyway, so this is the alpha, and so the bond from the nitrogen from one amino acid to the other does not bond to this alpha carbon. Sorry, alpha carbon. It binds to the carbon in the carboxyl group to the amino acid in the second with the nitrogen in the second amino acid. So this alpha carbon doesn't even get involved, but he keeps on wanting to call it an alpha bond. So, okay, I spent way too much time trying to explain this. Sorry. Let me go back. But basically, when he says alpha bond, he absolutely is incorrect. No microbiologist or biochemist would call that an alpha bond. They would call it a peptide bond. And we talked about this peptide bonds breaking. Oh, and then he thinks alpha bond is the correct way to bond. So what he's thinking is that for natural proteins, you have to have an alpha amino acid. So it needs to be peptide bonds between alpha amino acids. But he's conflating that with the bond. And that's why he keeps on saying alpha bond. And then the next is he thinks proteins are peptide polymers longer than 50 amino acids. So at 49 amino acids, it's not a protein. But as soon as you get to 50 and more, you get proteins. So that's another...
Remember, he called the short ones were enzymes and the big ones were proteins.
Right. So if it's 50 to some number, it's enzyme. But if it's like bigger than that, it's a protein. And I think by protein, he means maybe like a structural protein. I don't know, maybe collagen or something. So, it absolutely doesn't make sense. There are obviously huge enzymes that are very, very long, much longer than just a lot of structural proteins. And these are things like polymerases. They're huge. But you can also have a very small peptide polymer, just a few amino acids long that are enzymatic. I did look it up at one point, but short polymer enzyme. Actually, the best example is is insulin. Insulin is less than 50 amino acids, I think, and it's definitely an enzyme. So, it isn't because of the number of amino acids that it counts as an enzyme, or it counts as a protein, or it's only a peptide. So he's definitely wrong about that. But that's another example of where he's basically saying, oh, I mean, abiogenesis cannot happen because you would have to have, peptides that are at least 50 amino acids. And I didn't actually look this up, but I think in some of the experiments, some of the non-enzymatic polymerization of amino acids do go beyond 50 amino acids, or there are ways to get it to be 50, more than 50. But definitely you can have shorter ones that are still enzymatic. All right. Next, in terms of biological process, it says rotein, but it says protein. So we've been talking about this. He thinks protein cannot form outside the cell because a bond is too fragile. We know that's not the case. But the way he supports that is he brings up this chaperone protein. And he's claiming that these chaperone proteins form a bubble to protect the peptide bond. So you can just imagine like this cloud of small proteins that surround this larger protein. And the reason why it needs to be protected, of course, is he thinks that water, it will hit the bond and break it. But he thinks the chaperone protein are there to protect the peptide bonds. Of course, that's not the case. The chaperone proteins are to assist with folding of the proteins, especially when you get longer peptides with hydrophobic parts. They can kind of clump with each other. And what the chaperone protein does is they kind of keeps it separate until the protein fully forms. And in a sense that there are some cases, like I think especially in the bacteria, where smaller proteins can actually fold inside a cavity caused by the chaperone protein. But, that doesn't mean that the chaperone protein is somehow surrounding the protein at all times. Because if you did, that protein, that enzyme would not work. And so these are where he says he thinks abiogenesis has to create the chaperone proteins. Obviously not. And abiogenesis has to be able to create large proteins. No. He thinks ribozymes cannot self-reproduce because it's folded. So apparently the covalent peptide bond is too fragile, but the hydrogen bond and some of the other kind of weaker bonds that keep a ribozyme folded is somehow so strong that it's impossible to unfold. So that's why he's saying that ribozymes cannot reproduce. And of course, go ahead.
Except we know that the ribozymes unfold with temperature.
Yep.
That's all it's needed. And depending upon the structure of it, they'll unfold at different temperatures, like is seen in the one of the little archaeas that reproduce best at 100 degrees centigrade, which is remarkable that it can handle that, and that's its most comfortable temperature.
Yeah, being made of kind of structure made of RNA, that itself, tells you, or it would tell most people educated in this field, that the bonds that are holding that shape are fairly weak. And he thinks that RNA cannot reproduce, despite the citations. I think you had provided one. I had provided another one, where he just can't seem to understand, what the paper is saying, just like in the DNA barcode paper. So he keeps on saying that it's, there's no evidence, and and he keeps on asking for citations. But this is what kind of got me, and I commented to you, Blarg, about this. But when you gave him that one paper, his response was, well, that was a man-made experiment. So it's happening in vitro. So that makes it invalid from his point of view.
Yeah,
That
well,
pretty much says that he doesn't care.
Yeah, his argument was that that it's impossible to form outside a cell. Okay, here's a case where it's occurring outside a cell in glass, hence in vitro. And yeah, he then uses the standard escape hatch. You go, oh, well, that's not in nature, so therefore, and you just go, if it was impossible, then we couldn't do it either. So yeah, it's just a ridiculous argument. It's a fairly standard one, though, whenever someone's talking about abiogenesis. Like if we go along and create an organism from scratch using just straight abiogenic style of creation, they'd argue that, well, that's not real because that happened in a lab. So it doesn't prove anything. Yeah, standard escape hatch for all of these type of clowns.
And if you're going to essentially dismiss a research paper because, oh, it's man-made experiment, how can you call yourself well, not just scientists, how can you say that you actually accept science? You obviously don't accept science. Let's see. I've not been watching the comments, so I haven't been putting up some of the comments that people have made, and I see there's quite a few. I'll have to look past them later. So I'll keep looking out for now. So I
We were
guess you
just
guys
talking
are.
about we're just talking about prions in the comment section and how ridiculously hard they are to kill.
Yep.
Yeah, even sterilization in a standard autoclave doesn't do the job.
Yep. And the other thing that autoclaving doesn't do is it doesn't get rid of like RNase, which is an enzyme / protein. Just autoclaving it won't get rid of the RNase. And when you work with RNA, it's very difficult because RNase is actually very, it's pretty much everywhere. So you have to do some, I think, some chemical treatment to the, the different wares to get rid of the RNAs if you're going to work with RNA. I can't remember what some of those chemicals are, but yeah.
Miranda should be able to jump in on that because she said that that's the job she does for a living.
Oh,
Sterilize
okay.
surgical instruments for a living. And you just
Well,
go,
surgical instruments probably don't have to worry about RNAs. They're primarily worried about diseases, obviously including if there's prions, but it only comes into play if you are a researcher in RNA, where you need to make sure that the RNA that you work with doesn't. Neuro instruments get sterilized too unless a patient has a known or suspected prion disease. Yeah.
Okay. Yeah, that comes as a result of me asking whether if the surgical instruments come in contact with central nervous system tissue, is that the type that get thrown out immediately? And that's the answer to my question. Thanks, Miranda.
Yeah, and she did say they're not concerned about RNA. And Crystal made an interesting comment. So she says she knows nothing about biochem, but was able to look up various examples of RNA living for much longer than Ed's assertion of 20 minutes. Actual research papers. So in real life, RNA will degrade pretty quickly, like I said, because of the ubiquitous RNAs. But by itself, it's not going to just immediately just disintegrate in 20 minutes. All right. So then let's get to the rest here. Okay, things that RNA cannot things ribozymes can't self-replicate despite the research. And that was the research that you had provided. All right. Now, we
Certainly,
also,
certainly the research I had, it wasn't necessarily self-replication. It was just that they acted as a copying template. So it was a ribozyme that was able to copy other ribozymes. So yeah, so therefore it does that job that he said is impossible to do.
yeah, and he's, I guess to steelman his argument, I think he's talking about auto self-replication, right? Where basically you have um, an RNA, a ribozyme that reproduces its own code. But I think one of the other citations I gave did talk about that. Okay, next we
Yeah,
get to,
I'm not
yeah, go
sure
ahead.
whether the one I gave covers that or not. It's a little beyond my my reading level for that particular topic, but yes, it certainly did talk about it being able to replicate RNA molecules or polymerize RNA molecules using a
Yeah,
template.
basically, yeah. So basically, you don't need to go to DNA, at least not right away. You can have RNA ribozymes and use ribozyme as a template. So, and the theory is that once you get to that point, you then move on to DNA as a template because DNA is going to be more stable than RNA. And when I say DNA is more stable, single-strand DNA isn't that much more stable than RNA, but definitely double-stranded DNA is more stable. Okay, so we talked about him not understanding half-life. Oh, and the other thing. So he thinks not being found in nature means that it's impossible, which logically doesn't make sense. Just to say that something hasn't been found isn't some proof that it's impossible. But he keeps on saying that.
Well, jet aircraft aren't found in nature, so apparently they're impossible. The point
Uh,
I'd make though there with the specifics to do with abiogenesis is that we wouldn't expect really early types of small autocatalytic sets to exist in the modern era because there's no way those are going to be as efficient as the really complicated ones which have evolved over the 3 billion years or so, nearly 4 billion years since the first organisms come on the planet. So the moment something like that tried to exist, it would just be turned into food for something else immediately. So yeah, we're never going to find anything like that because everything, every surface on the entire planet is covered by something that will eat it.
yeah, life has gotten past the point where you need that self-replicating RNA.
Or where it can even exist,
Yeah.
because of life. Yeah, would cull it immediately. It has no chance, and which is why we also don't see a second abiogenesis. We've got one line of life that creates one group of animals and one line of life that creates a completely different one. Whichever got going first is the one that has the leg up, and everything that comes along behind it, it's a paper plane trying to fight against fighter jets. It just has no chance.
Where it could happen is if it's in some environment that's completely separated from everything else. So let's say like deep underground, there's some place where there's no other microbes, no real microbes, but there's enough, conditions that might let it occur. So you could get a biogenesis occurring, but it would take so long that and it would have to be in a place that would last that long. So it's almost a near impossibility that it would happen again. Not
Yeah,
that
it
it
can't get out in the
is,
wild,
yeah,
basically. So we won't see, one group of organisms roaming the plains of Africa that start from one abiogenic ancestor, and then a completely different set of organisms to start from a different one roaming the same plains. That can't exist.
But Blarg, you're completely wrong, because, you know, fish had to walk out of the ocean with a fully formed functioning legs, according to Ed's theory of Pokemon evolution. He actually says that.
Yeah.
Clark, did you actually see that video of with Dr. Dan and
No, I didn't.
Oh, okay, yeah, it's more of the same. It was
It's the
just
same.
completely frustrating.
The same garbage we get from Nepi as well, saying that unless all of these organs evolve simultaneously 100% in one go, then it's impossible. And you just go, well, that's utter garbage, because there are so many different types of fish that have, for example, lungs. I've got a little fish on my kitchen bench that has a completely different style of air breathing capability, and is in fact an obligate air breather. It will drown if it can't get access to the surface. And that's it's a fish with lungfish, of course. There's also the arowanas and those giant fish in the Amazon that are also obligate air breathers that will drown if they can't get access to the surface. And their their gills are essentially only used in the way our kidneys are for getting rid of nitrogenous waste. So, you know, these are things that exist. There's also, for example, tube worms that don't have any stomachs of any kind. They have no opening, no gastrointestinal tract of any kind when they become adults. But according to Nepi, these things all have to exist or an organism can't live. And you go, well, that's we see examples where that's not true, and we see examples of the evolutionary steps along the way all over the place. For example, the lungs in lungfish.
Yeah, and those are the things that you would predict. That you would predict these type of animals with these type of structures, if you know these organs evolved over time. You can't do that with creationism. You would have to say you would have to ask the question of why would a creator design these kind of intermediate organs and organisms? What would the purpose be?
Yeah, they can't live on the surface and they can't live underwater entirely. They have to live at the boundary between the two, or otherwise they will die.
Yeah, you have to definitely watch that video. It's
yeah.
all right.
I'll
So
put it on the watch list.
yeah. Okay. So things that okay, and so if not being found in nature means that it's impossible, then God is not found in nature, not that I've seen. So it's impossible for him to exist, according to his thinking. And he seems to have this, and I've ran across this with him from the very beginning when I first ran across him on some of the other channels, where you ask him to cite something and he can't cite it normally. Eventually he gets to the point where he might give like the title of the paper or might mention the author, but he can't give like a formal citing. And also he like over and over, he can't quote anything. Whenever you ask him to kind of quote what, whatever is in the paper that backs up what you said, and he can't. So, he doesn't seem to have had any academic background.
Yeah, the irony of that is, remember the list of demands he had? If someone says no to something that he says, you've got to provide the paper.
Yep.
And then you just go, when it comes to him, you just go, oh, no, everybody obviously knows that.
Yeah, he does a lot of that, right? Oh, you don't need a paper because, everyone knows that. It's obvious.
But that list that he gave you of demands is almost a carbon copy of the one that James Tour hands to people. So, more pure if he's a fanboy.
I definitely noted that these two behave in extremely similar ways.
Even the
Highly
body
aggressive.
language.
Yep, highly aggressive. But the difference is James Tour actually should know better.
Yes. Well,
because that guy genuinely has an amazing education.
Yeah, but I mean, you become a Massey on it, Jew, and it's you have to have had a lobotomy, as far as I'm concerned.
So Aaron Massa made that type of comment under that video. He says, "This is James Tour scale stupid. This has me waiting for you to yell, Mr. QC, at the top of your lung."
Write it on the
If you've seen,
chalkboard.
yeah. If anyone's seen that debate with Professor Dave, that's just like,
priceless.
oh my God.
Did you see the video also with James Tour sitting with a whole bunch of actual researchers?
Yep.
That was embarrassing. That was a little bit like the embarrassment, that essentially in the same scale of embarrassment as the of the video of Trump's daughter trying to talk to some world leaders, and they just look at her like she's a moron and move on with the conversation. And you just go, ooh, you feel embarrassed on behalf of them. It's so palpable.
That's my next one to watch. I was up early this morning. I did a sort of dive through trying to find old stuff of Ed's and Chewers, and I found that one and stuck it on the to watch list. But Ed has been in the comments of the AXP before. I just got to correlate. My files got slightly confused. I ended up sticking some of his comments in with one of Apot's new bloody names or whatever or something. So I've got to sort of go through and pull it out. But yeah, he's
Do you know
commented
about
before.
when he was commenting?
Give me a minute. Talk amongst
Yeah.
yourselves.
Because, I haven't seen his comments, I think, or maybe I didn't notice, at least for a few months. Oh, so Tuna Man, is this stuff get nuts and futs? Is that? Oh, no, that's the Apot, right? I think someone said that.
Yeah, that's him. That's that. Also known as Nathaniel Elderwood was the first account he came on with. Where he told us he'd just got out of jail. And then was very cagey about it, and there was some rather suspicious stuff about the way he talked about what it was that he was in jail for. And then after a couple of months, he came up with a story that it was to do with selling weed, I guess.
And
that's probably not true.
That was his second time in jail. The first time he was done for stolen vehicle, grand theft. Or at least stolen vehicle, I remember that.
Yeah. If you're
Nathaniel.
cagey, go ahead.
Yeah. I should also note, for those who are listening, there isn't another person who is called Nathan or Nathaniel who pops up occasionally, who's definitely not the same person. He's the fella who's in his mid to late 20s, who's quite severely autistic. And you'll be able to spot him because he has really short, like, couple of word sentences, like the devil is evil and God is wonderful, and he does those type of comments. He's poor bastard is not. He wasn't given a very fair chance in life because he already has some significant challenges, and he's clearly been heavily indoctrinated into this. So even when you point out that he's lying about these, another one he says is that the that KKK are atheists,
Oh,
and
yeah.
you just go, well, no, you have to be a Christian to join the KKK. It's a part of the requirement, and you can show him that, and then he'll just go on and say it again. So yeah, he's not honest, but he's also not the full package. So I generally cut him a little bit more slack than some of the other trolls. So that's a different guy. If you come across another guy by the name of Nathaniel, and you're getting those type of comments.
Yeah, and for Apot, now that I know who that it's him, I try not to even engage him, at least not directly. But I was just gonna say that if you're cagey about the reason why you went to jail, it's not because you were selling weed or you stole a car.
He claimed he rented the car, but he forgot to take it back, or some story like that. And the amount of weed he got caught with, he was caught in a state where it is illegal, and I think the amount he got caught with was trafficking amount. From memory, I'd have to go back, but yeah, I did find where, yes, Edlen542. I don't know how long ago it was. I think at the end of last year, but he came on something. He started up with how did evolution build the eye, the optic nerve, and the portion of brain that it needed to. And I just said, well, box jellyfish have got basic eyes, and they don't even have a brain. So what's your point? I think they can even find primitive eyes on some algae or something, or at least light sensitive things.
Actually, the latest Z Frank video. If you haven't seen Z Frank doing his vids, they're terrifically entertaining. But the latest one actually talks about the eyes on jellyfish. It's about jellyfish, and on box jellyfish in particular, they have a little stalk that has an actual sort of compound eye on the end of it, plus a couple of other little sorts of eye spots. But and it has a little weight in it so that the eyeball remains upright all the time.
So in the body of the
in the body of the little eye stalk.
Yeah, especially when you look at the Irriganges, because it's so small. If you look at it under magnification, you can actually see quite clearly the black dots. I think there's 4 sets of them. And yeah, they just seem to. A professor at James Cook in Cairns does all the work with the Irriganges, and he was trying. Last I heard, he was trying to work out how it stays orientated up all the time, because it's not the bell isn't really a flotation device in the Irriganges, because it can go against the current and actively hunt. So yeah, I must catch up with that. And what was the name of the channel, mate?
It's in the comments. ZEFRANK. Cool. I'm typing it out again. But also, if you look also for the one that has octopus in it. There is a bit of a video of an octopus there walking along the bottom of the seafloor. Every time I see that thing, it just breaks my frickin' brain. I just refuses to be part of what it is that I'm looking at. You go, it's one of the most remarkable things I've ever seen. It's insane. Yeah, brain just wants to crawl out my ear and run off because that is not how octopus move. Yeah, so yeah, Z Frank, brilliant channel.
Is that the video where like the octopus is stiff-legged?
Yeah, walking like a person along the seafloor.
I caught some of that.
Going, what the actual is that? And also the octopus that has got a coconut shell or a clam shell that it sits in and walks along with. And you're going, these things are insane.
As portable camouflage or something. I don't know.
Oh, yeah. Camouflage is amazing. But yeah, he's got a great channel that terrifically funny, but also extremely good quality information. And he puts all of the papers that he's referencing in the at the end of the video as well. So so it's all coming from actual proper research. So that's good too.
Yeah, I mean, the eye is such a good example of evolution. It's funny when these guys bring up the eye to say that it's irreducibly complex and therefore evolution isn't true.
I mean, correct me if I'm wrong, but I thought the mantis shrimp had the most complex eye so far that we've found, that humans have found.
Not quite. In some ways, yes. In some ways, not. So they have gargantuan, what do you call it, segmented eyes, like most arthropods. They have something around about, from memory, 13 different color receptors. But that doesn't give them 13. It doesn't multiply their ability to look at color compared to us, because that's not how they use it. So essentially, if anyone's had a look at the way in which the James Webb Space Telescope does some of its work, it's got a couple of filters that it can apply to an image from something so there'll be about 10 or 12 of these filters, and at some point you'll see the star disappears, and from there on you can't see any of it. So that's a little bit how this works. It's just they have it's just an on and off rather than a blending of all of the colors. So they just essentially see that one color for that one receptor. So yeah, they have a lot more colors, but we actually can probably see more color than they can in total. Well, more different colors. We can't see out into the infrared and ultraviolet the way they can. So they've got detectors certainly out into the ultraviolet as well. But yeah, those little beasties are insane as well.
I can never understand why anyone would even want to try and put their finger close to one to see how, do they really break the sound barrier when they punch?
Oh yeah, they cause cavitation.
Yeah, they do.
They crack crab shells with cavitation.
Yeah, but I mean, let's go put my finger near it, and you'll see the dive of basically all these bubbles coming out from the respirator.
And you see stupid people also when they've got them in a tank, in a glass tank, put their finger over or put some food over near the glass, and they can smash the glass on the tank.
But you'd have to keep them in the tank. I thought you would use bulletproof, that sort of type glass.
Perspex. Make it out of Perspex. It can withstand the impact without shattering.
Yeah, I didn't. Yeah, my mate, he made all the housings for the AIMS, the Marine Institute. Did the same thing. He made it all out of it. Yeah, silly me. Okie dokie.
All right.
Sorry, we've derailed your conversation, Casey.
It's all right. Yeah, I get self-conscious if I'm talking the whole time during the session. So there's a good kind of breakaway. But I think we're almost done here. We did talk about him dismissing citations because they refer to man-made experiments. And here's another example. What was that?
Penguins.
Oh,
Penguins.
no, I think that comes up. Yeah, that comes up at the end. So he thinks that for 40 years, they were trying to find ribozyme replication in nature when it clearly says that they were focused on synthetic ribozyme. Um, so this is an example of him just like completely like misreading what papers say. And we saw that.
That's an example of him completely quoting James Tour, because that's the same shit that Tour says. They've been trying to find it in nature, and it's
Yeah,
no.
so this one was in regard to a citation I gave. So what he does is that he doesn't actually try to read and understand the paper. He tries to he searches probably for some keywords, and he comes to some place where he thinks that it confirms whatever he thinks is. So in this case, the paper is talking about how like we said, we're not going to find self-reproducing RNAs in nature as of now, just because it's not going to survive long enough for us to detect it. And there are no organisms that are going to still be using that because it's so much more inefficient than the systems that are available now. So we know, I mean, it's not unheard or it's not surprising that they couldn't find that in nature. But what the paper says is that, you know, uh, it hasn't been found in nature. So during the last 40 years, researchers have therefore focused on the engineering of synthetic RNA systems that self-reproduce. So there, this doesn't say that they were looking for 40 years to trying to find this. They pretty much said, hey, there are no such things in nature that we know of. So let's focus on trying to synthetically do this to demonstrate that it is possible, that there are kind of ways that you could have synthetic RNAs that reproduce. And ironically, this paper goes on about the actual different methods that people have found to demonstrate that RNA can self-reproduce. So there are just many systems here that's being mentioned. And so instead of like actually looking at this and understanding what this means, he thinks that the papers is confirming what he's saying, that RNA cannot self-reproduce, when the paper itself is showing RNA self-reproducing. Okay. All right. So yeah, that's the final one. So he does a lot of kind of miscommunication. So he thinks that I blocked him. There's no evidence that I've blocked him. And the fact that he still has comments on here pretty much says that I haven't blocked him. So then he kind of went into this, well, it's because you've reported my comments. And what happens is that if, let me see if those go into, let me go over to here. So if I do report him, so if I go here and say report, and let's say I say it's misinformation, and I report, what I get is, no, this is too small. So I get the option to hide the user. It's too bad it doesn't show up here. So it's not the reporting itself that causes the block for him to be not. Let me go through this again. Where is he? So I think when I reported, his comment may disappear, but and you can see that I have the option to hide the user from the channel. And when I do, all his comments would disappear. But if I go to report misinformation, this is all display is all messed up.
Yeah.
Because I'm trying to
Can't scroll down far enough.
Yeah. And it's because I'm doing this in a way that, but anyway, so when I do that, what happens is when I report, I get the option to not hide that. At that point, I get the option to hide the user. So if I select on hide the user, it'll be same as if I just directly went and selected hide the user. So just reporting itself isn't going to stop him from posting anymore. It's just going to possibly remove his the reported comment. Although I think if it may just come back, it just kind of disappears momentarily. So even that isn't valid, I guess. Let me see if I can do this. So if I report and I say misinformation, and when I click on report, oh yeah, so then you can't see it here, but never mind. I'll give up on that. So anyway, yeah,
it doesn't remove the comment.
Yeah, but basically, I haven't blocked him. I haven't done anything of that kind. And one of the things that I did want to discuss here is to kind of what to do with him, because he's just saying stuff that is adding nothing, and he's not writing anything new. It's just the same stuff over and over, just continuing on to just keep saying his ignorant comments and incorrect comments.
Read the blocking. Ed doesn't understand, and Blug and I have had both of us have actually had this happen to our accounts.
What do you mean?
If you get reported enough times,
yeah,
YouTube will red flag your comments, and they will eventually, your comments will stop, just be shadow banned continuously. And
well, first you get like you get put on timeout.
Yeah,
you can't post anything for like 24 hours.
That doesn't even happen. That doesn't necessarily even happen. So my other account, all the comments are visible from the other account, but are not visible to anyone else, no matter how. So normally, if you're ghosted, if you go and sort the if you go and sort the comments by newest first, you're able to see all the ghosted comments. My first early account, you can no longer see anything that I've ever written. And that was without having, I did once have the account banned completely and terminated, but it was restored like the next day, because it was done by a malicious person with about a dozen accounts of their own. So I'm not sure what the report was for, but anyway, I got an instant termination, and then it was recovered, and all my comments were returned as normal. But yeah, at some point, all of a sudden, they just stopped showing up and are no longer visible on any channel, basically. So, and I never got any more warnings. There's nothing there. It's just something really odd.
Yeah, it's exactly the same thing that happened to me.
Yeah, there's no path to getting it restored.
No, they won't answer your emails. I've had my old account suspended, I think 3 times, and restored virtually within 7 hours or the next day, with all the comments showing up again. And just like blood, bang. The only way I can see comments from my old account is to actually log on to that account and go and look for them. And so the gods of YouTube are even more mysterious than any Christian version of God.
So just to confirm, so here's the settings where I have the hidden user. So if I blocked someone, it gets counted as a hidden user, and they will appear here. I haven't blocked anyone. So for him to just continually say that without any evidence that that's the case, pretty much says that he's a liar. He's completely dishonest, and he's just saying things just to say things, and actually doesn't care about what the truth is. That's basically where we are with this guy. So the question comes back to, what do we do with him, in the sense that I don't want to hide him, because it's going to get rid of all the stupid comments he's made that I think at some level is entertaining, and it kind of demonstrates how bad some of these guys get. So I don't want it all to disappear. But, he is just kind of disruptive. And for that reason, he's been banned or blocked from a lot of channels, including apparently Aaron Ra's channel, because he created an account that says Aaron Ra blocks you and Dave blocks you. I don't know if they've actually blocked him, or he's just being stupid.
That's on my list to do investigations when I can't be vertical anymore. Yeah, so I'll have a look at that one. Read what to do with him. I'd say leave all the comments that he's made so far up. But if he decides to start commenting on like this video, or if you make any more this live stream, or any of your videos or anything, I'd say just remove his comments.
Yeah, and I think you guys as moderators can delete his comments. Blarg, you said you can, right?
Yeah. They're not deleted per se. They're popped into another section. Yeah,
okay.
in for review. So I have done that to two of his comments.
Okay.
Yeah, so definitely can. So yeah, if you were to go over into your account and have a look at that, for that, you'd see the sum total of everything that's been done like that as well. So and I'm gonna guess it's just those two that I removed, but yeah, I'm on the same page as Ook. But of course, this is your channel.
Yeah, I haven't done anything, removed any comments, even though either of Ed's or other people's, but I've sort of come close and said, no, I don't know how that works. I'd have to check. So whether QE can go back in and reinstate them.
So let me show you. So these are the comments that are held for review, and this says removed by moderators. So I think this is probably one of the ones that you removed, right?
Yes, those two. Yep.
Okay. Yeah. So what I would say is that I would recommend everyone who's a moderator on this channel to feel free for any future comments that he makes where you think that is completely useless or abusive or whatever. Go ahead and do that. I mean, I'll review it, and if I disagree, I'll put it back. But I doubt it'll be worthwhile to do that. So if you guys want to do that, any of you guys who are moderators, feel free to do that for any new comments. I think we can leave the stuff he's said, some of them, even if it's abusive, to leave those. But any new ones, completely feel free to delete his comments.
Yeah,
that's the way I sort of looked at it as well.
Yeah, I've looked at all his comments, and they could be seen as abusive or bullying, but I don't. If they had, I look at it this way: if they were in, say, one of the Lines comment sections or the AXP comment section, I wouldn't bother reporting them, because I don't think they technically break any of YouTube's TOSs, and that's the only reason I report anyone.
He is running 5 accounts now.
Yeah, apparently that is allowed.
Yeah, but 5 demonstrates its troll behavior.
Yeah,
so he's
multiple. That's a huge one. Yeah.
And one of them was called Run QC, is the brand new one he just created.
So, because I'm obviously running from him, right?
Yeah.
Oh, it looks like Diva, I yanked his chain and he ran away again.
Oh, Diva?
Yeah, I warned him. The first and last warning, I type in all caps.
Oh, okay.
Ran away.
But so, the other thing he does is, and I told him, like, if you really want to test whether you are blocked, all you have to do is just type something innocuous like test, something that the YouTube filter wouldn't get. And then if it shows up, he's not blocked. So he's continued to do this, like test, despite the fact that, he can make comments. So if he can already make comments and it shows up, he doesn't need to put test. This is just like stupid level stupid.
Well, actually, it's him trying to validate his accusations, trying to show evidence that, look, I have to test whether I can comment every time.
But the thing is, like, his regular comments under this account shows up, so he's not being blocked.
I don't think he knows how YouTube really works, the comment section.
He might not. And his claim that this is showing up is because I've blocked all his other accounts, but somehow this slipped, he was able to slip this past me, because as soon as he put 542, I couldn't figure out who that was.
Just scroll through the others, though, because if they're blocked, we wouldn't be able to see any of the other account names.
Yeah.
So, and you just go, I'm pretty sure we can see the other account names. So there's one. Look, Ron QC.
Yeah. I mean, if we go back even to the other one where he started, like
there's
this
the
one,
other
8632.
I
mean, they're all there.
Just double checked. He's running 7. He's used 7 so far on this on QC's channel, different profiles.
Yeah.
So because I hadn't noticed the 2 Ed Gain ones, and then the brand new one for this thread, which was Patrick.
Yeah, the Patrick is the got it open here.
Well, it comes up in the comments as Patrick, but has he commented as Patrick?
The Patrick profile shows up as Run QC.
Okay, yeah.
If you click on it.
Okay.
Yeah, in the live chats it shows like the name that you put in and not the handle. So
that's
Miranda
an annoying thing.
Yeah, I think so.
Just give us the damn name, not the handle.
Yeah,
if we want to know
just be
what the handle
consistent.
is. Yeah.
So Miranda says that his comments can get buried on big channels like AXP. Yeah, here is, and this was a case with the Underlings channel, and he obviously has a lot more subscribers than me, but just from sheer volume, he just would post so many things that, even if it's relatively big channel, it'll still show up. So it's just a matter of how much effort he spends on this. So I think we're agreed with in terms of how to deal with him moving forward. So I think that's good.
Yeah, I'm in the buy section. Yep.
And so I've saved the best for the last. He doesn't know that penguins have feathers, and that's because he says he's not an expert on penguins, but
they didn't
he's
teach
somehow
that in college.
Well, I mean, he he's not an expert in biochem or microbiology or philosophy or any of those things, and he still comments on them.
Sorry, hang on.
But I think you know enough about penguins after grade school that, or maybe junior high, where you can reasonably guess that they do have feathers.
Well, he admitted on Dr. Dan, the Creationist, Dr. Dan. He said he went into a big thing about all birds are classified as this, and their things are the beak and the feathers, and then he went to say penguins don't have feathers. So there is no bird in the world that has no feathers, unless it has a disease or maltreatment or something like that. So yeah, but yeah, to use the excuse they never taught us about penguins in college is just insane. And when I mentioned the marsupials. And what about the monotremes? He said the monotremes just Pokemon evolution, they just popped out fully formed. I mean, the platypus, perfect example. It produces milk, lays eggs, but it has no nipples. The milk just comes out from its skin. A marsupial, when it's born, is less than the size of a gummy bear. And it just finishes gestation in the pouch. I mean, it's a perfect example of how evolution progresses. Yet, nope. Doesn't work. Crazy.
And by the way, the fact that the milk just comes out in the skin actually points to how the mammary gland evolved.
Yes,
I think,
yeah. And it's interesting thing, they found fossilized jaw bones of not platypus, but of extinct platypus, and they actually have teeth, functioning teeth. So again, now it's just if you ever stick your finger in a platypus's beak, well, one, make sure it's a female, but two, it's just like it's like sort of a weird little squishy thing. You can't. There's not even any real pressure. Yeah, but the extinct platypus, they found with jawbones with the teeth in them, and this is only vaguely remembering. I think they go back to the start of the Australian megafauna, so the start of the marsupial megafauna age in Australia, which is, remind me, blog, 60, 80 million years ago. Or yeah. Oh, I can't remember. Too many painkillers. Brain's not quite working.
Sorry, what's that, Ook? How far back?
When did the great mammalian megafauna in Australia start?
Oh, yeah, no, not real sure. I know they survived up until basically humans arrived.
Yeah, till the first immigration wave of what we now call Aboriginals. Yeah. Yeah.
Yeah.
No, I'm drawing a blank on how long, but yeah. But I mean, back then we had ducks that were the size of kangaroos, wombats that were the size of hippopotamus or slightly bigger. Yeah, he has absolutely no idea of how evolution works, but Ed's whole point to his argument is because you don't know the exact process, how it started, everything we know about evolution is false. And yeah, it's
a fallacy they keep bringing up where if you don't know this, if science doesn't know X, then God must exist or there must be a creator.
Yeah. I mean, it's ridiculous.
If he was ever to be honest, I guarantee you he is a creationist. He might be an old Earth creationist or whatever the Messianic Jews believe. I have no idea. What was the other new one that that troll, the new troll who Jackie, the repeat caller who got banned from all the line shows, then moved over to Talk Even, and now it's moved on to the line. Comes in with all the 16-year-old. He's a 16-year-old who uses the big philosophy words. Jackie is a Noahide, and they're Christian-ish, but they're Gentiles who follow the 7 messianic laws of Judaism. So that's a weird one. I'd never heard of that one before. I think Bristol will be able to spell it. I couldn't spell it, but yeah, Noah Hyde. But that's what Jackie is, the big waffler, or Gishgal, the god or creation. Weird. I just love
So
it.
Blarg, oh sorry,
Yeah,
Blarg,
mate.
what did you mean? What did you mean? Penguins are extinct? I didn't get the joke.
Sorry, I missed that bit.
Yeah, grab my next comment as well. Pop that up. There it is. That is the original thing called a penguin, and that bird is extinct. It's also known as the great auk. It looks like a penguin. Yeah, it's a northern hemisphere bird that looks like a penguin, lived like a penguin. It's a swimming bird, but it was hunted to extinction in the 19th century to make pillows and mattresses and stuff out of.
Because they had feathers? Is that what you're saying?
Well, yeah, because their feathers were so downy and wonderful. Yeah. And then explorers discovered penguin-looking things down on the down in the southern hemisphere and went, "Oh, those are penguins, but they're not related." So ironically, actual penguins, the original thing that was called penguins, has become extinct, and then an entirely unrelated set of birds that have a similar lifestyle and look similar have taken up the name penguin.
That's actually very interesting. I didn't know about that.
Yeah, and the same thing happened with pandas, because red pandas were the original thing called pandas. Yeah. It comes from a Nepali word. And then Westerners discovered the things we call giant pandas and went, oh, they look similar, so called them pandas and the other thing red pandas, but they're not, they are sort of related, but not very closely as well. But yeah, the things we now think of if someone says panda is not the original panda either. Hilarious.
Yeah. Nice. Okay. All right. So I think then we're done here. I think we're pretty clear what we need to do with Ed. And I mean, we don't necessarily have to remove every comment he makes, but if he's being particularly stupid or obnoxious, then like I said, feel free to remove his comments. All right. Yep. So I think
I have returned.
All right. So I think we're done here. Miranda asked, so the cutesy cartoon penguins at the North Pole in Christmas pictures aren't so far off. Yeah.
Yeah, maybe at one time. I don't think they actually went up that far, but yeah, there were definitely northern hemisphere penguins.
That's a good one. Yeah, I think you've covered all the points. I got nothing else to add.
All right, then we will actually end relatively on time. Normally we seem to go past the 1:30 hour 30 mark, but thanks everyone. This was very fun rehashing Ed. Oh, by the way, so in terms of the session, the special session on Monday, what did you guys think? Is this something that for future is worthwhile doing, or are guys like Ed just not worthwhile?
Situational, depends. I mean, we didn't really have an eye, or you had an inkling of what Ed was like, but you gave him a chance. And I mean, most people deserve a chance. But I mean, doing a special one, that's a big ask.
And what was that? What did Krista say?
She's just making comments about David's posting in the comment in the chat. Yeah. I don't think you need to go out of your way unless you particularly want to. But yeah, I mean, you bent over backwards for him, and I don't think you're obligated to do that. But again, up to yourself. But if you do run another one on a Monday, most of us will, time zone permitting, try and be in for it.
Yeah, but it makes it more difficult on a weekday.
Yeah, my take is if there's people available, fine. It added, essentially, it's a data point for those people who may be considering either going back to theism or they're hovering at the edge of theism, wanting to leave, and then they can look at clowns like this and just go, you know what, that's just embarrassing, and that may well be, that tiny little bit that helps them get over the edge. So yeah, it's not necessarily a bad thing to add a moron to have a conversation, a belligerent moron to have a conversation and demonstrate how little he actually knows and how dishonest he really is. But yeah, it's certainly it's one of the ones where you do need to put a stomp in him to stop the carrying on. So
all right.
Well,
yeah. Good show, Q.
I appreciate everyone's feedback, and we'll end it here. Thanks everyone in the live chats for your participation. It always makes things more interesting or entertaining. So I appreciate you guys being there, and I appreciate you guys on the stage for coming on. And I will end it here, and I'll see everyone in the comments.